Okay, so we've talked at some length about all of these rules of inheritance that Mendel worked out. And we ended with the idea that there's some sort of little elements, some kind of particles, some sort of physical thingies rattling around inside cells that we now call genes that are responsible for the traits that organisms have. But can we get deeper into that black box? What are these gene things? And we have to jump back in time to see how this was solved, back to the 1600s, when a gentleman from London by the name of Robert Hooke was trying out an invention of his called the compound microscope. You can see it looks a little bit different from the ones that you have used in lab, but the basic idea is the same. You put several magnifying lenses, one in front of the other, and use their combined power to magnify objects. Hooke actually published a book of things that he'd observed called Micrographia that was a bestseller of the time. People just couldn't get enough of his drawings of, what fleas and flies and such really did look like up close. And one of the things that he did look at was slices of cork. That's the inner bark of a particular species of oak tree called the cork bark oak, sensibly enough. All trees, as far as I know, have a similar layer in their bark. What liquid is it holding? You mean this? If you mean this diagram of the microscope, if you mean that thing over there at the left, let me see, check in the chat. What liquid is it holding? No, K. I'm not entirely sure what that's all about. What liquid is the letter K holding? Oh, I see. Okay, I think that's not a vessel of liquid, although I can see how it looks like that. That's actually a polished piece of metal. Without electric lights, what Hooke had to do for illumination was light a candle and then put a reflector back at the candle to reflect as much light as possible into that lens, which is the big round thing that you're seeing, and then shine as much light as possible onto what he was looking at. So I don't think that's a vessel of liquid. I think that's a reflector drawn at an interesting angle. I could be wrong. But yeah, it's all part of how you lit up specimens back before the invention of electricity. Anyway, so Hooke drew this slice of cork, and the chambers that he saw in that cork reminded him of the cells in a monastery, so he called these things cells. Now, as far as heredity goes, people had known for quite some time the basics of how babies get made. You and your preferred partner, make whoopee together, and 9 months later, you get a little bundle of joy. The cellular basis of the process took a little bit longer to figure out. Human sperm cells were first seen in 1677. It took much longer to find egg cells because human eggs aren't produced in nearly as many numbers, and human egg cells are also much harder to get to since they're internal. An early idea was that either egg cells or possibly the sperm cells actually contain tiny little duplicates of the parent. This is a sketch of a sperm cell that somebody claimed he'd actually seen under the microscope with a tiny baby folded up inside the head of the sperm cell. And then presumably this tiny baby, if it's a male, has testicles which contain sperm, which contain tiny little babies inside them, and they must contain sperm with tiny babies inside them, and so on. So basically all of humanity is, nested one generation within the next, kind of like the ultimate set of Russian dolls. That's not the way heredity works. And it just as well, if every sperm cell contains a tiny little baby, which will grow into a full-grown baby and then an adult, given the right conditions, if that's true, then, every healthy teenage boy kills about 100 million babies a day, sometimes twice a day. But fortunately, this is basically false. This is not how we get, this is not how we reproduce. This is not how heredity works. So if we skip a couple of centuries up to 1839,
you're right, I'm gonna be at work first.
If you could mute whoever that is. Theodor Schwann and Matthias Schleiden, a couple of Germans, were the first to propose that all living things were made up of cells. Several generations of microscopists had found cells elsewhere, not just in cork, but in animal tissues, plant tissues, pretty much everywhere.
Love you. Love you.
And Sierra, if you could mute, please. And they stated that what defined a cell was having a nucleus. And yes, this leaves out prokaryotes. That was something that later had to be modified, but let's not get into that too much. There's some drawings that they made of various kinds of plant and animal cell. And as Schwann put it, talking about plants, though the variety in the external structure of plants is great, their internal structure is simple. This extraordinary range in form is due only to a variation in the fitting together of elementary structures, which indeed are subject to modification, but are essentially identical. If you look at the buildings on campus, they have many different shapes and many different functions. The student center, the library, the chapel, the Lewis Science Center, the dorms, etc. They look different from each other and they do very different things, but they're made out of a limited number of the same kinds of subunits. They're all made out of brick. They're all made out of drywall and timbers and a limited number of components of the electrical and the HVAC and things like that. There's a fairly limited number of different types of part. It's just that you can combine the parts in different patterns to make anything from a small chapel to a dormitory to the new Integrated Health Sciences Center and that arts building that they're putting up on the corner of Donaghy, right? So there's a limited number of parts, put them together in different ways and you can make anything. The same is true, Schleiden says, about cells. There's a limited number of cell types, put them together in different ways and you can build anything from a rose bush to a redwood to a corn stalk to a blade of grass. Schleiden and Schwann had originally thought that cells might crystallize out from some sort of living fluid, some sort of goo that contained the power of life. But after some more years of careful observations, Rudolf Virchow in 1855 stated that the only place a cell could come from was another cell. Omnis cellula e cellula, all cells from cells. By the way, I might have a couple of test questions that involve you remembering some of these names, but the only thing I would ask for would be last names. Don't memorize dates. If you've got to prioritize, focus less on the people and more on the ideas. It's more important to know about cells than to know how to spell Schleiden. Skip up to the 1870s, microscopes have gotten a lot better. There are limits derived from the laws of physics as to how much detail you can see with light. Even the finest light microscopes imaginable cannot resolve things smaller than about half a micron. And that's ultimately got to do with the nature of light. And microscopes in the 1840s, and certainly the earlier ones that people like Robert Hooke had worked with, weren't nearly good enough to start pushing those limits. But by about the 1870s, microscopes were in skilled hands pretty much as good as yours are, possibly better. And it was getting easier to observe cells and also to observe structures inside cells. And this is going to seem irrelevant at first, but in 1856, an English chemist by the name of William Perkin was doing chemical analyses on byproducts of coal and discovered that one of those byproducts was a vibrant purple in color. This was the first invention of artificial dyes. That purple, by the way, was called mauve. That is a silk dress dyed with this first artificial dye, mauve. And many more were soon discovered after that. And this is ultimately the reason why you can buy clothes in just about any color you can think of. Instead of having to dye your clothing with plant dyes, which could be expensive, it was possible to make all kinds of artificial dyes very, very cheaply. The reason we care about this is that some of these artificial dyes that were discovered after 1856 turned out to be very useful in looking at cells under the microscope. Not only do they increase the contrast, many cells are pretty much colorless, and under the microscope, they're just translucent and you can't see a lot. But dyes, or as they're usually called in biology, stains, make it possible to see transparent structures, and some of them can pick out certain structures, but not others. One of the artificial dyes that was discovered in the 19th century, by the way, is one that you've actually used in lab last week. It's Sudan 4. And you might remember that that stains in the presence of fats or lipids. So you could zap a cell with some Sudan 4, and the parts of the cell that were rich in lipids would stain strongly, but the parts of the cell that weren't rich in lipids would not stain very strongly. So you can pick out different areas of the cell based on what they're made of. Oh, another dye that was invented at about this time that you've also used is methylene blue. I'm sure you remember that, and you'll be using that again a little bit farther on in the course. Okay, so not only do artificial dyes make your clothing a lot more colorful, a lot of those stains turned out to be useful in looking at cells. And in 1873, a guy named Schneider was looking at cells in the tail of salamander tadpoles, if memory serves. And he was the first to observe something rather unusual about eukaryotic cell nuclei. Normally, over on the left, you can see what an ordinary cell looks like in the tail of a salamander tadpole. It's kind of a roundish cell, and the nucleus looks like a round, grainy blob. But cells divide. Remember, every cell comes from a previous cell, and the way you get more cells is by the cell dividing. You started out as a single fertilized egg cell, which divided into 2 to 4, then 8, then 16, then 32, ... until we ended up with the roughly 37 trillion cells that make up you as an adult. And Schneider noticed that as a cell divided, the nucleus stopped looking like a round, grainy blob and looked like it turned into a set of little stringy objects. And the stringy objects moved in some kind of pattern. And then the cell divided, and the stringy objects turned back into round, grainy blobs. And these are his drawings of that. At the left, that is a cell as it looks most of the time. And then here is a cell about to divide, and you can see these little stringy things in the middle. I'm highlighting them for you. And now you can see the little stringy things that look like they're getting pulled to opposite sides. And now here we have 2 sets of stringy things that have been pulled apart. And you can see that the cell is beginning to pinch itself apart, and will soon pinch off into 2 new cells. And the reason I brought up that whole business about artificial dyes is that those stringy things turn out that many stains would color them very strongly. If you don't stain the cells, they are virtually transparent. But if you do add some stain, often the little stringy things take up the color very strongly. And you can see there's a lot of contrast between the stringy things and the rest of the cell. Now, we can't just going around calling these objects stringy things. So they came to be called colored bodies, which in Greek is chromosomes. You may have heard of chromosomes. Well, this is what they are. They are these stringy things that appear and move around when one cell divides into 2, and this is what they look like. This picture was taken by a type of microscope that wasn't available in the 1800s. It's called a scanning electron microscope. So this is jumping the head a little bit, but this picture shows what it is possible to see under the light microscope is that when cell division begins, when a cell begins to divide and the chromosomes appear, each chromosome is made up of 2 parallel strands, and we call those strands chromatids. So if you look at this, this is one chromosome right here. This is one chromatid. And then this that I'm circling is the other chromatid. Right there, almost like 2 hot dogs that are stuck together in the middle. And then that right there, ooh, look, computer animation. Whoops. And then that chromosome right there is also made up of 2 chromatids that are stuck together in the middle. So at the beginning of cell division, each chromosome kind of looks a little like an X. So there's the 2 chromatids. And each chromosome that has 2 chromatids, they're connected at a spot called the centromere, right here and right there, more or less. That's the centromere. Mere, by the way, comes from the Latin root meaning a division, and centro means the center. So the central division of the chromosome, where the two chromatids are connected, is called the centromere. Now, here's the thing. When a cell is not dividing, you can't see the chromosomes. When a cell is not dividing, if you looked at it under the microscope, the chromosomes would just look like or the nucleus just looks like this grainy blob, and you cannot pick out individual chromosomes. But we have reason to think that the chromosomes are still there, even though when a cell is not dividing, you can't see them. One of the things that was noticed pretty soon is that the number of chromosomes stays the same from one generation to the next. If you have a cell that's got 20 chromosomes when it divides, if you let it divide and then you let the two daughter cells live and grow and then count chromosomes when the daughter cells divide, each one will have 20 chromosomes. That number 20 stays the same from one generation of cells to the next. What's more, the number of chromosomes is characteristic of a species, with two very important exceptions that we'll get to later, probably Friday. Virtually all normal human cells have 46 chromosomes. And in fact, they come in 23 pairs, a fact which will be important later. Right now, this is a picture of human chromosomes that have been stained with some rather colorful fluorescent stains. And if you care to count, you'll get 46 chromosomes in the count. And that's true of all normal humans. That with the exception of eggs and sperm, which I'll talk about later, and things like red blood cells, which when they're mature lose their nuclei, every one of your cells that's got a nucleus, turns out inside that nucleus there are 46 chromosomes. On the other hand, chimps would have 48. Mice, I think, would have 40. Horses, I believe it's 64. Different species have different numbers. The number's not really that important, but different species do have different numbers. And that odd little set of movements that chromosomes go through when cells divide has a name. It's called mitosis. Mito happens to be Greek for thread. So mitosis means threadosis, I guess, might be how you would translate it. There we go. Now, this may seem like learning about cell division, there's some stages that it goes through that you have to be familiar with. And this might seem like it's a bunch of trivia. One of the points that I want to make is we've already covered Mendel's laws. What you're going to see if you stick with this is that the discoveries that Mendel made and the discoveries that various microscope jockeys made in the 1870s and 1880s about chromosomes turn out to be opposite sides of the same jigsaw puzzle. This may seem like a bunch of pointless trivia to memorize right now, but it's ultimately part of a much bigger picture. And getting to the heart of that bigger picture is going to be very rewarding if you stick with it. So I'm not giving you this level of detail because I absolutely love to cram your brains with stuff you don't need. I'm giving you pieces of a bigger jigsaw puzzle. And what I want you to try to do in this class is assemble those pieces. And we will soon get to where we can see how everything I've been talking about connects up. Okay, with that, what you're looking at is a cell in, this happens to be a fish embryo that happens to be a type of cell that shows cell division pretty clearly. And this is what a cell looks like in interphase. The nucleus just kind of looks grainy, and chromosomes are present, but you can't see them. And the cell is just hanging out. It's doing its metabolism. It may be growing. It may be, doing whatever it needs to stay alive. It's, probably talking with other cells on its cell phone, because cells are connected by a cellular network. Right, sorry. And we call this interphase. When a cell begins to divide, never mind for the moment how a cell, quote, knows when it's time to start cell division. I might be able to hit that a little bit later, but don't worry about it for right now. When a cell begins to divide, usually the membrane that separated the nucleus from the rest of the cell, called the nuclear membrane, disappears, and the chromosomes start becoming visible. This is another one of those fish embryo cells. And you can see that in the center, you're starting to get some blobs. This cell, by the way, has been stained with a stain, possibly methylene blue, that picks out the chromosomes very well. So in life, the chromosomes would not look blue, but because of the stain, you can see them more clearly, and you can see that they are in the process of becoming visible. When chromosomes become visible, we call it prophase. Also in prophase is something going on you can't really see in the picture that I showed you, but you can see it here. What you're looking at is a cell that's been photographed with a special technique called fluorescence microscopy. We can actually do this here at UCA. We've got it's called a confocal microscope that can make fluorescent pictures like this. And the chromosomes have been stained so that using this type of microscope, they fluoresce in blue. So all of this electric blue mass right here, that is the chromosomes. And two tiny little organelles that are called the centrosomes, like right there and right there, produce a set of fibers inside the cell, microscopic fibers. The fibers are called spindle fibers, and the structure that they make is called the mitotic spindle. You can think of the centrosomes and the fibers as being the motor and cables that actually move chromosomes around. Chromosomes cannot move on their own. They have to be pulled. And specialized fibers inside a cell give a cell its ability to move things around internally. We don't really need to get into how that works. But these special fibers do the work of moving chromosomes, and they appear in prophase. And the fibers here under fluorescence microscopy have been stained to glow a rather fetching bright green color. So the green is showing you the fibers. The blue mass right there, that's the actual chromosomes themselves. Okay, once the chromosomes appear, they line up in the center along an imaginary plane that cuts the cell in half. The plane doesn't really exist. It's just a plane that bisects the cell, and it's called the metaphase plate. And you can see that the chromosomes here have lined up in the center along this imaginary line. That imaginary line right there is the metaphase plate. It's the spindle fibers that are moving them into place. And in this particular picture, if you look carefully, there and there, those two tiny dots, those are the centrosomes. And if you really squint and probably take some really good drugs, there and there and there and there, you can kind of, if you use your imagination, see those spindle fibers right there. So you can slightly see this under the light microscope. But here's what it looks like under fluorescence. The chromosomes are this blue goo in the middle, and the bright green fuzzy stuff right there, those are the spindle fibers with the two centrosomes kind of at the centers right there of those star-shaped masses of spindle fibers. And by the way, when this happens, when the chromosomes line up in the middle, we are in what's called metaphase. So the chromosomes line up in center on this imaginary plane called the metaphase plate, moved into place by the spindle fibers. Where things start really happening is in the next phase called anaphase. And in anaphase, the chromosomes break apart at the centromeres, and separate chromatids move to opposite sides of the cell. So each chromosome did consist of two chromatids that were stuck together. It's at anaphase that the centromere breaks and each separate chromatid moves to opposite sides of the cell. And here's where you're seeing that happen. Right there and right there, we have the centrosomes. You can, if you squint and probably take some really, really good recreational neurochemicals, you can faintly see the spindle fibers. And those two separate lumpy dark blue blobs right there, each of those is separated chromatids that have broken apart at the centromere and are now in the process of being pulled to opposite sides of the cell. And that's anaphase. And there's what it looks like under fluorescence. Again, these blue areas right there, those are the separated chromatids of the old chromosomes. And in green, you can see those spindle fibers. All right, checking the chat. Can I go back to the last slide? Certainly. Right, so that's what anaphase looks like under the light microscope. Remember that when we started mitosis, the cell division process, each chromosome was made up of two chromatids that are joined together at the centromere. What's happening here is that centromere has cracked in two, and each chromatid is now getting pulled in opposite directions by the spindle fibers. Okay, all right, you may have noticed I'm not a world-class computer artist. This is the best I can do on the fly. But we started with each chromosome made of two chromatids, and in anaphase, the chromatids separate from each other, and each chromatid that made up a chromosome is pulled to opposite sides of the cell. And again, we call that anaphase. All right, can I continue?
Yes, you're good.
Okay. And again, that's what it looks like under fluorescence microscopy, which shows you the spindle fibers much more clearly. And then finally, the separated chromatids on opposite poles of the cell cluster into new nuclei, and they start losing their individuality. It looks kind of like they're dissolving. They turn back into a grainy blob, and you can't see it clearly here, but a new membrane is forming around each new nucleus. And we call that telophase. Telos is Greek for a goal. So telophase is the phase that was the goal of all of this mess. And incidentally, all the phases we've talked about, interphase, prophase, metaphase, anaphase, telophase, which you are going to, or which you really need to be able to remember and recognize in order, all of those stages technically apply only to the nucleus. Usually, the cell itself, the whole thing, divides when the nucleus is in telophase, and the division of the entire cell is called cytokinesis. Kinesis is Greek for movement. Some of you might be majoring in kinesiology, the study of human movement. A situation that's kinetic is one where things are moving. And cyto is the root for cell. So cytokinesis is movement of the entire cell. And usually cytokinesis happens at the same time that telophase does. If it doesn't, then you end up with a cell that's got two nuclei, which it would be pretty unusual in human tissues, but there are some organisms where that can happen, and you can have cells with two or often more than two nuclei. We don't really need to get into that. But usually at the same time when you have telophase going on, the entire cell will pinch in two, kind of like, twisting a balloon in two, and you end up with two separated cells. So telophase is what the nucleus is doing. Cytokinesis is the division of the entire cell, and usually they happen at the same time. And there's the spindle fibers again in telophase. You can see that we've now got two very distinct clumps of chromosomes, and each one of those will go on to form a new nucleus, and the entire cell will pinch off in two and will end up with something like that. Okay, just checking the chat. Incidentally, cytokinesis looks a little bit different in plant cells. In animal cells, usually when cytokinesis happens, the entire cell just pinches in two. A little bit like if you just took a soft candy and pulled it apart and twisted it a bit, you could get it to pinch apart into two pieces. Plant cells division looks a little bit different because plant cells have a cell wall, and that can't pinch apart because it's solid. What happens in plant cells is that a new cell wall will form to divide the cell in two. So this is a plant cell in telophase. These two red masses there are the masses of separated chromosomes. They are going to go on to form new nuclei. And faintly visible right here is the beginnings of a new cell wall that's going to partition the cell into two cells. So much like if we were remodeling a building, you can't take one room and squeeze it to make two. If you had a room and you needed two rooms, you just build a partition wall in the middle. Same thing with plant cells, which can't really pinch in two because they're surrounded by that rigid wall. Okay, that's actually a pretty good place to stop because we are at 10:50, and I will go ahead and stop the recording. And I'll stop it now.